And with that, it is my great pleasure to introduce Dr. Siavash Kurdestani, who is a full professor and chair of biological chemistry at UCLA. He got his medical degree from Harvard, and then he went on to do a postdoc in Michael Grunstein's lab at UCLA, and he received a UCLA Chancellor's Award for postdoctoral fellows, and he joined the department in 2004. And as I mentioned, he's now professor and chair of biological chemistry. I'm really excited to hear about the evolutionary history of histones and eukaryotes. I've been a big fan of Dr. Kurdestani's work for a while, especially linking metabolic processes to chromatin and how chromatin can actually behave as, like, I think you call it a metabolic organelle, which I think is a really cool way of saying it. Yeah, with that, I'm going to leave the floor to Dr. Kurdestani.
Thank you so much, Christine. Thank you for your generous introduction, and thanks to Kelsey for a great talk, and also the participants who are here for the talk. Let me share my screen. You guys can see the slideshow, right? Okay. I'm going to talk about this project that was just published a couple of months ago. And because all the sort of primary data is in there, I will sort of briefly talk about how we came to work on this project, some of the issues that we ran into, and then I'll wrap up by telling you a little bit about the current and future projects, showing a little bit of unpublished data. Although I have to say, everything that we've had, we put in that paper, so I don't have too much unpublished data. Whatever we have is from the last couple of months, given the COVID situation. So we started thinking about this project, really sort of thinking about what was the function of histones in the first place in archaeal species. This is because archaeal histones form a structure that's very similar to the H3H4, and obviously the eukaryotic histones derive from these archaeal histone molecules. But these histones occur in organisms with small genomes, and obviously they're prokaryotes, so they don't have a nucleus, and they lack N-terminal tails and even the enzymes that you typically modify histones. So the typical answers for genome compaction and gene regulation that we invoke for why we have histones didn't seem to apply as much to these archaeal species. So this sort of raised the question whether histones have an additional function that served as the reason for which they're evolved, and that function may have been conserved in today's eukaryotes. In trying to think about this question, obviously this is very difficult because, where do you start? And so we did, every so often we try to go back and read about something different that we work on and try to see whether we can come up with hints and clues as to what histones may be doing. And eventually we came across the literature of eukaryogenesis, and specifically the changes that were happening in the chemistry of the planet at the time. So what I'm showing you here is on the x-axis is time, as billions of years ago, and what you see is that obviously life initially evolved in an anoxic environment, and a little over 2 billion years ago, oxygenic photosynthesis was essentially invented or evolved, and this resulted in generation of oxygen, which just started to interact with everything and oxidize, sort of molecules that it came in contact with. So just like today, where we have global warming, a little over 2 billion years ago, we started having global oxidation. And what happened at the time, among the many changes, was that many of the metals that were really abundant in the ocean, and they were in the reduced form, such as iron and copper, either directly or indirectly as a result of interacting with oxygen, became oxidized or bound to other molecules and sort of precipitated out of the ocean. And in fact, this resulted in so-called these banded iron formations, which is essentially rust or iron oxide. And these layers sort of correspond to increasing levels of oxygen. And so what happened is that essentially iron disappeared from the face of the planet. There were also other changes in other metals, although the chemistry there is a little bit more complicated than iron itself. And it was in the middle of this sort of mess that sort of eukaryotes appeared, right? And the question was, whether there was something special about this, endosymbiotic relationship or this species that allowed it to survive in such an environment. And so, given the coincidence of appearance of eukaryotes and this changes in the geochemistry of the planet, especially in the metals, we started sort of thinking about whether there's a relationship between the histones and metals. And we actually spent a lot of time working on iron because that seemed to be the biggest change. But over time, both in our thinking about it and also some experimental data suggested that copper is really the metal we should be looking at. So copper is an essential element in eukaryotes, and it's used by many enzymes and pathways. What's important is that copper, however it's brought to inside the cell, it has to be in a reduced form to be chaperoned to its destination proteins. This is because as I mentioned, our biochemistry evolved in an anoxic environment, and so much of our metabolic pathways had to adapt to the presence of oxygen. And so most of our chaperones had evolved prior to appearance of oxygen, and so they were capable of binding copper-1, not copper-2, and this remains the case in today's eukaryotes. Two major pathways or activities of the cell that depend on copper are obviously mitochondrial respiration, where copper is delivered to the complex 4, cytochrome C oxidase, and copper is the last metal center that electrons go through before they get to the oxygen. SOD1, or superoxide dismutase, which is involved in oxidant defense, also has a copper, and it receives copper from a chaperone that brings copper-1 to it. Once copper is in the destination protein, it may function as in a copper-2 or a copper-1 state, but it must be in the reduced form, or what we call the biousable form, for it to be trafficked to the destination protein. Now, we thought that for this protomitochondria to have survive the initial state of endosymbiotic, the host cell must have had a mechanism to get copper to the mitochondria, or the mitochondria must have had a mechanism to hijack copper from the archaeal host. But this suggested that this archaeal host must have had a mechanism to maintain copper in its reduced form and be able to traffic it intracellularly. Now, this just remains a hypothesis, but this is how we sort of came to think about whether histones may have played a role in that. So really the question for us was, could histones have aided the emergence of eukaryotes by providing biousable copper to the protomitochondria, which would establish essentially one arm of this endosymbiotic relationship. And doing a little bit of literature search, it turns out that people actually had identified a few metal binding sites in histones. And the one that caught our attention was a region between histone H3 proteins that form the interface that holds the H3, H4 tetramer together. So the H3 is shown here in copper color, and the H4 is in blue. And all the interactions that maintain the H4, H3, H4 tetramer are within this region. And as I said, this was important for us because this is the ancestral form of the histones. And so we thought that if something's happening, it should be at this particular metal binding site. And there are a pair of histidines and cysteines at this site that form essentially what looks like a copper-2 binding site. This is very similar to other copper-2 binding sites in other proteins. So the question that we asked was, could this be a copper reductase enzyme to catalyze the reduction of copper? We obviously discussed at length in the lab whether it had to be an enzyme, whether this was just a binding site for copper-2, and that could potentially explain some stuff. But you know, it didn't seem that the binding would make sense. Like, we couldn't come up with a hypothesis that would explain some of the phenotypes that we're seeing in our yeast cells that I'll show you in a couple of slides. That could be explained by just binding alone. And so catalysis was something that seemed crazy but plausible for some of our phenotypes. So to answer this question, we had to come up with an assay that can detect a copper reductase reaction, which is shown here. Basically, you start with copper in the +2 state, and you have an electron donor that transfers electron to copper-2 to generate copper-1. The assay that we developed that actually took a long time, dependent on these chelators that specifically bind copper-1, and then they turn blue or yellow, so you can actually detect the color, and by measuring absorbance, you can follow the progress of the reaction. So I'm going to show you a master video production by my iPhone, probably 4 at the time, where I have basically a buffer control, the H3, H4 tetramer in this well, and unassembled H3 in this well here. H4, when it's unassembled, it's insoluble, and so we couldn't do a reaction with it. And what you'll see is that I will actually add copper to this reaction and look for a change in color in the H3, H4 tetramer well. So as you can see, nothing happened, okay? And so when something like this doesn't work, you don't know where to begin, right? Do you have the correct assay? Do you have the correct tetramer? Do you even have a correct idea? And all of these questions become more existential as time goes by, because it really took us almost 3 years and more than 150 tetramers later to be able to actually get this reaction to work. We, for a long time, had no idea what the problem was, right? So we worked on the assay for a few months. We tried to think about if there's something wrong with our histones. So we isolated histones from bacteria, playing classical music for them, trying to be gentle, and none of that stuff worked. At some point, I felt like I'm on Titanic, and I actually have information about the fact that it's going to hit an iceberg and sink, and I'm still like staying on Titanic. And I'm happy to talk about this afterwards, but anyway, so it turns out that the main problem for us was that there was a lot of contaminating metals in our buffers that we normally use in the lab, and so we had to decontaminate everything: glassware, chemicals, FPLC columns, all the reaction tubes, and essentially everything we used, and that resulted in generation of active tetramers. So here's a repeat of that reaction. Here we have the BCA chelator for copper 1. And so we expected this to turn bluish purple color. And as you can see, the reaction is a little bit slower here for the purpose of presentation, but you can see that over time, this turns purple, suggesting that copper was being reduced, copper 2 was being reduced to copper 1. This is what it looks like when the reaction is done. And just like any other enzymatic reaction, you really don't change the equilibrium. It's just like how fast you get there. So if you let this plate sit here overnight in a little over 24 hours, these other wells also show similar color. And you can follow the progress of this change in color in a little cuvette and get quantitative data. So this is the amount of reduced copper that you get as a function of time in seconds shown here. This is what you get in a buffer for a tetramer. This is a reaction that occurs with the tetramer. We're missing the early time points here because the reaction is super fast in these conditions. And as soon as we add copper, while we're pipetting, the reaction is happening. And so the first reliable time point that we get is 2.2 seconds after addition of copper. We've done a lot of controls, such as mutating the tetramer when the activity goes away. If you boil it, the activity goes away. And we've measured enzyme kinetics. In these conditions, it's a decent enzyme. It reduces about 800 copper 2 ions per second. I have to say, even though we put in a lot of effort to try to get good enzyme kinetics, I'm not sure if any of this apply to in vivo conditions because copper must be brought to the tetramer or the nucleosome with certain chaperones, and copper 1 must be taken away with other chaperones, and in absence of those chaperones, it's hard to tell whether these kinetics are the same in vitro and in vivo. So, but this is what we get given these conditions. Now, if we mutate the histidine to various residues like asparagine, tyrosine, or alanine, that affects the activity of the enzyme, and so suggesting that the histidine 113 is required for optimal enzyme activity. Now, we were actually doing experiments in parallel in vivo, trying to understand whether this copper reductase activity is relevant. And we also used the Saccharomyces cerevisiae as a model system, where we generated a mutant strain with both copies of histone H3 mutated at histidine 113 to an asparagine. We initially tried to make H113 to A or alanine, which is a typical mutation that you make, but the cells are not viable, and this has actually been reported in the literature in the past. And but we looked in cancer databases, and we noticed that histidine 113 is actually mutated in certain cancers, including in melanoma, to asparagine. And so we thought maybe this is a mutation that the cells can tolerate, and that's why we sort of focused on this particular mutation. Our genetics showed that this is a hypomorphic allele, and but I want to show you the data here. But we wanted to basically see now if this mutation affects copper levels, especially copper 1 levels inside the cell. For that, we developed a transcription-based reporter based on transcription factors that modulate target gene expression based on presence of copper 1 or copper 2. I'm showing the reporter here that senses copper 1. This is a transcription factor that activates its cognate promoter, and we put that in behind the GFP to get a GFP signal. We showed that this transporter works really well. So in YPD shown here, you get a certain distribution of GFP signal in your cell population. As you add increasing amounts of copper, you get increasing amounts of signal. And if you deplete copper by adding a chelator, that decreases the signal. When we looked at our mutant, it was clearly it sort of shifted to a lower levels of signal, indicating that there is much less copper-1 in this mutant compared to our wild type. And this was associated with copper-dependent defects inside the cell. I'll show you one example here, which is respiratory growth. So we took our cells with wild-type histone H3 and the mutant histone H3. We deleted the CTR1 copper transporter to essentially zero out the levels of copper inside the cell, so we can add increasing amounts of copper to detect the difference between these two mutants. So at very low levels of copper, you can see that neither of the strains grows well, because in absence of the copper transporter, the copper levels inside the cell is very low. If you add a little bit more copper, now the cells with the wild-type histone H3 grow, but not the ones with the mutated histone H3. And in fact, it takes much more copper to rescue this growth defect, suggesting that really the defect has to do with copper levels or copper usage inside the cell. There's a ton of data in the paper. We used extensive genetics growth assay. We measured oxygen consumption directly. We measured cytochrome C oxidase activity directly through other means other than oxygen consumption, to show that our histone mutants affect respiration in a copper-dependent way. We also looked at SOD1 activity, again, through extensive genetics growth assay, assays for copper-1 loading into SOD1, as well as enzyme activity, all of which indicated that in this histone mutation, you have defects in SOD1 activity and mitochondrial respiration. Now, what I didn't tell you at the beginning was that when we started this project, we had noticed that histone H3 was missing this crucial cysteine in some fungi, including Saccharomyces cerevisiae. Now, at the time, we didn't quite realize the importance of this cysteine, and so we started our project in this organism, which we've used a lot. And it turns out that this actually caused quite a bit of problem for in vitro assays, because we could not get the in vitro wild-type yeast H3 to show any reduction. We're still not sure why that's the case. We think that really has to do with the way we assemble the yeast tetramers. They don't assemble as well, but it could be also to other reasons. But for some evolutionary reasons, some fungi have lost this, the cysteine, and there's alanine there. Now, there's precedence for the fact that the cysteine can be absent in certain copper proteins because its function can be substituted by hydrophobicity. And I'm happy to talk about this more later. But the absence of this cysteine allowed us to actually now use it as potentially a gain of function and in our experimental condition. And so we have now generated a mutant that has a substitution in alanine 110 with the cysteine. Now this restores the activity of the tetramer in vitro. In this context, if we mutate the histidine, that decreases the activity. And when we look at this strain again in vivo, it has more copper-1 based on the reporter. Again, this is we thought it was a very strong evidence because it's a gain of function mutation. And very satisfyingly, this presence of the cysteine actually rescued the lethality that's associated with H113 to alanine mutation, suggesting that perhaps some problems with the H113A could be insufficient copper reduction inside the cell. That addition of the cysteine completely rescued the growth defect and in fact lethality of this mutant. One last piece of data I'll show you, which I really like, is that this mutant is very good at using copper. So in oxidative condition, in presence of potassium cyanide, which poisons the electron transport chain, both of these strains have problems growing. But when you add a little bit of copper, you can see that this A110-2C mutant just takes off because it's much better at using very little amounts of copper that's in the media compared to wild type. So we thought that this provides a very strong evidence that the yeast histones are also functioning as copper reductases inside the cell. So we think that histones are intimately involved in maintaining copper in its biousable form inside the cell, and that they distribute it to various destinations inside the cell and potentially outside the cell in multicellular organisms. So I wanted to just take a couple of minutes to tell you about some of the projects that are ongoing in the lab. We're interested in looking at the structure of the H3H4 tetramer from the perspective of an enzyme. And that makes you look at this structure very, very differently. And some residues that would not capture your attention at all, such as leucine 126, become a very sort of important residues in the catalysis. So this particular residue actually is near the copper binding site. It actually forms a cage, and the hydrophobicity of this residue is actually is important in other copper binding proteins in terms of changing the reduction potential of copper, or the ability of copper to accept electrons. And so we actually have generated mutants of L126 where we've made it more hydrophobic. For example, we put a phenylalanine there. And you can see that this actually acts as a major gain of function mutation where compared to wild type in relatively low amounts of copper, it shows a growth advantage, which then goes away when you have abundant amount of copper. We're also interested in the activity of the nucleosome, potentially role of remodeling complexes in regulating this activity, post-translational modifications, and histone variants, both the variants of H3 as well as variants of other histones that are incorporated in the nucleosome. Now, people assume that the nucleosome should be active, but we're not sure if that's the case because it's unclear what roles H2A and H2B play in this process. Now, H2A has a lot of variants, and there's also some very interesting residue changes between these variants, and I'm highlighting one here, where if you look into the active site from like sort of inside the nucleosome with two different H2As, which some of them have an arginine at position 99 and some of them have a glycine at position 99. You can see how this arginine versus glycine sort of affects the sort of the structure and potential accessibility of this region to copper and maybe other cofactors. And in fact, in yeast, when we delete one copy of H2A and H2B, we actually get an increase in copper 1, suggesting that presence of H2A may actually be inhibitory to the nucleosome. Although we have to rule out other explanations for this change, including transcriptional changes. And so, so please take this with a grain of salt, but I wanted to say that, you know, a nucleosome in its full form may or may not be active. And in fact, removal of H2A may be necessary for bringing about this activity. Finally, we're interested in how this activity is linked to other cellular biology, especially copper homeostasis, redox state of the cell, and potentially diseases that the histone enzyme activity may contribute to their pathogenesis. We're focusing on diseases with iron sulfur cluster dysfunction and some cancers in which histones are mutated. The reason for this is that copper, especially copper 1, is toxic to many proteins, including proteins that include iron sulfur clusters as cofactors. And copper itself can be actually very toxic to mitochondria. And so we think that there must be a reciprocal regulatory mechanism between mitochondria and the nucleus, specifically histones, that control the production of copper and weighs the sort of the cost of generating copper against its toxicity and consumption of reducing power to generate copper 1. Okay, I'm going to finish by acknowledging important people that contributed to this work. This work was led by Narcis, Oscar, Maria, and Chen. Narcis and Oscar were graduate students at the time, and Maria Chen was a postdoc, and Maria is a project scientist. I'm indebted forever for their contribution to this project, for believing in it and sticking with it, and also constantly challenging me and each other to sort of think deeper and harder about some of the issues that we had to solve. We got a lot of help from Mike Carey, who's our neighbor, and as well as Scipio Merchant and his postdoc, her postdoc, Stefan, who were at UCLA at the time, but they've moved to UC Berkeley. And I'm going to stop here and ask and entertain questions. Thank you so much.
Amazing talk, and also just such an amazing example of perseverance in science and really getting to the bottom of these questions. Oh, actually, okay, so Tobias Wernicke already has his hand up, so go ahead and let him talk. Okay.
Hiya. Given your framing, I was wondering whether you checked the archaeal histone sequences and whether they would be, any of them, would be consistent with the same functionality.
Yeah, so the archaeal species, that histidine is highly conserved in essentially all histones, and I think in most, if not all, archaeal histones. They don't have the cysteine. We're trying to see whether these archaeal histone species have activity or not. It may be very difficult to do, but we're trying. The reason it's difficult is because, establishing electron transport chain in water, or electron transport in water, is very difficult. The electron transport chain occurs in the membrane because you need hydrophobicity, because essentially water short circuits your electron transfer. And so with these proteins that lack cysteine, we think the hydrophobicity is important. And so it might take a little bit of time to find the right conditions in vitro in which we don't disrupt the hydrophobicity and allow the protein to reduce copper. Now, I have to say that if that turns out not to be the case, then we were extremely lucky, because we basically, our reasoning led us to this hypothesis and this discovery. And so we would have to think about how to walk back essentially from now this data to archaeal histones.
So we have a couple questions from Pallavi Mustafi. 2 questions, yeah. So she first says, amazing talk, very naive question. Do you think mitochondrial DNA may also form such active tetramers?
No, mitochondrial DNA actually don't have histones. I mean, they're clearly bound by other proteins, but they're not chromatinized. And
yeah. Oh, sorry, go on.
And prokaryotes are very careful about use of copper. I actually don't know of any copper binding protein inside the cell. They either put copper from outside into the complex 4, or they bring it in through very, very strict chaperones and then stick it into the complex 4 or other proteins that are in the membrane, because copper is extremely damaging to DNA, especially if it's not bound to histones.
Yeah, and then her next question was, do you think this enzyme activity may affect the nucleosome, may have any effect on nucleosome binding and altering its structure? I think she means like maybe other factors binding to nucleosomes.
Yeah, so as I mentioned, sort of everything is fair game at this point, right? It's hard to know which activity is related to this, which is not. So if it turns out that H2A and H2B are actually inhibitory, and as most of you know, H2 and H2B are rapidly exchanging in chromatin, right? That may be a mechanism for controlling the activity, and that then opens up the possibility for all the chaperones that are involved in that process to play a role here. We have some evidence that chaperones do play a role, but whether it's direct or indirect, it remains to be established.
So Alan Underhill also has his hand raised, so go ahead and let him talk.
That's great. I have a copy of your paper that's essentially covered in yellow highlighting. I was just wondering, like, how close is that particular active site, the reductase site, to the surface of the nucleosome, and can it contribute to modulating the activity of other chromatin modifying enzymes, a lot of which are actually poor enzymes on their own?
Yeah, this is something that we've thought about. I don't have a sort of a strong evidence to give you, but it's a possibility. It's definitely accessible, and in fact, the C-terminal region of H2A it sort of comes very close to that site, but yeah, I mean, there must be some interactions with chaperones and potentially even DNA itself. It's possible. These are good questions. I just don't have a good answer for them.
It's great. It's beautiful work.
Thank you.
We also have a question from Daphne Augusti, and she asks, do you think that viruses, which have also been around for millennia, may have contributed to the evolution of histone copper sensitivity?
That's a very good question, and in fact, Daphne, during Kelsey's talk, that's all I was thinking. So I'll email you some ideas I have, and maybe we can start a collaboration. It certainly occurred to us that viruses may want to control this activity, again, because as I mentioned, copper can be extremely damaging to unprotected DNA or genetic material. And so it's possible that viruses control this activity, either repress it or even activate it under certain conditions if it benefits them. So that's certainly a possibility.
Daphne.
Hi, I just wanted to, I'm here, and I'll say we can talk more in the coffee hour if you're going to hang around a little bit as well.
Yes,
that'd be great.
Yes.
Definitely. Cool. Alan, I think, are you raising your hand again to ask another question? Go ahead if you are. Okay.
No, sorry. Okay.
Okay, sorry, sometimes I get confused with the hands raised. My bad. We have another question from Patrick Varga Weiss, and he asks, do other ions also bind at this site and get reduced?
Oh, very, very good question. So we've looked at zinc and nickel and cobalt. They don't bind, or they don't bind at low concentration. You may have to go to very, very high concentration to detect some binding. So we don't think they're sort of natural substrates. What binds very sort of competitively with copper is silver, which is just below copper in the same periodic table of elements. So it has very similar orbital structure, and so it was expected for silver to bind, and in fact it does. It doesn't get reduced. We have also looked at iron, and iron doesn't get reduced at all. We actually initially thought that this may be an iron reductase given the absence of iron, and we spent maybe a year trying to work out an iron reductase condition. And even though chemically we can see iron reduction, so we think our assay works, and it's very similar to the copper reductase reaction. We've never detected iron reduction consistently in the same conditions as we look at copper. But this raises the issue of whether the older alkyl histones may be involved in iron or copper reduction as well, and so that remains a possibility.